Yes, hello Pasangala, Vanakkam and welcome. A very good evening to all of you. Yes, a very good evening. யாரெல்லாம் லைவ் பார்த்துட்டு இருக்கீங்களோ ஒரு green hearts கொடுங்க. Bio green hearts கொடுங்க லைவ்ல. Let's start the live. The most important live நிறைய பேர் கேட்டுட்டு இருந்த ஒரு live. Most important diagrams. I'm just not doing diagram. I'm also doing diagram based questions pasangla. So hello, I'm having many people who are joining in. A very good evening. எல்லாரும் எப்படி இருக்கீங்க? How are you all? Yes, hello, yes. Yeah, biology-நாலே diagrams எவ்வளோ முக்கியம் கரெக்டா? ஸோ உங்க CBSE-ல diagrams மட்டும் கிடையாது பசங்களா. You will be getting diagram based questions also. Good evening, Abhinaya. Hi, Sam. Hi, Shruti. Hi. A very good evening, everyone. Yes, voice is low. How is the voice now? Ipo okay va? Is it fine now? Ipo better? I will try to be a little more louder. No, this is for class 12. This is for class 12. Class 12 biology. Yes. CBSE. Okay, fine. Shall we get into the session? Romba exciting ana session. Okay, chapter wise. Okay, from chapter 1 till the last chapter, I have actually curated the most important diagram and also the most important diagram based questions. Yes, we shall get into the session. Okay, yes, my voice is still low. Is it still low? Am I not audible still? Is it fine now? Okay, start pannalam. The very first lesson, okay, உங்களுக்கு reproduction in organism இல்ல, so எல்லாருக்குமே தெரியும். We have sexual reproduction. Voice is not, hello ma'am, it is not low. Okay, great. ஆனா இன்னும் கொஞ்சம் ஜோரா பேச ட்ரை பண்றோம். Okay, I'll be a little more louder. Okay, so first we have sexual reproduction in flowering plants, the most important diagram based chapter. நிறைய diagrams இருக்கு. It is for 12th. Okay, I will be even more specific. Class 12 CBSE 2024 is the target. Okay, yeah. முதல்ல கொடுத்திருக்க diagram என்ன பசங்களா? Can anyone tell me what is this diagram? So அதுக்கு தான் நான் headings கொடுக்கல. So let's try to interact with you guys and find out what is the heading in பார்த்தேன். Yes, I am Nivetha. I am Nivetha. Okay, I'm Nivetha, your biology master teacher. So I also teach on NEET Tamil channel. I'm basically a NEET educator and I also teach for 11th and 12th CBSE. Okay, yes. The first thing is, this is an anther wall. Section of anther wall, very important. Okay, hi Elamati, hi. Okay, so you have, your anther wall has 4 different regions or 4 different layers. The outermost epidermis, அதுக்குள்ள endothelium. Okay, happy lori, yes. நம்ம தமிழ்நாட்டுல போகின்னு சொல்லுவோம், happy bogi everyone. Okay, then we have middle layers and the innermost microspore mother cell and we have a tapetum. இந்த 3 layer இருக்குல்ல, உங்க epidermis, endothelium and your middle layer, they are going to help in protection. Okay, and the tapetum is going to help in nourishment of the developing pollen grains. Yes, so in the picture அப்படியே ஒரு screenshot எடுத்துக்கோங்க. Yes, this is a very important picture. Yeah, I teach only NCERT. Yes, that is CBSE. Right now I'm teaching only CBSE. Okay, yeah. Next, pollen grain. So what is pollen grain? It is a male gametophyte. Okay, so in the pollen grain la rendu idu add பண்ணிக்கோங்க labelings. Okay, so this is your exine, this is your intine. Okay, this is my vegetative cell and this is my generative cell. So rendu cell irko, onnu vandu vegetative cell. Hi, all of you joining in. Hi. Okay, one is your vegetative cell, other one is a generative cell. இப்போ ஒரு question கேக்குறேன். In the vegetative cell, generative cell, which cell is going to develop into male gametes? இந்த ரெண்டு செல்லுல ஒரு செல் மேல் gametes develop ஆகும். அது என்ன செல் பசங்களா? Can anyone tell me which cell develops into male gamete? It is your generative cell. Yes, very good. So it's a generative cell that develops into male gametes. They can ask you, பாருங்க biologyல diagram கேட்டாதான் எழுதணும்னு கிடையாது. If they ask you, explain the structure of pollen grain ன்னு சொன்னாலே, it is understood that you guys should draw the diagram. ஸோ டயக்ராம் எழுதுனா என்ன தெரியும் அந்த எக்ஸாமினருக்கு, சரிப்பா இந்த பசங்களுக்கு எல்லாம் புரிஞ்சிருக்கு, நம்ம ஃபுல் மார்க்ஸ் அவங்களுக்கு கொடுத்துடணும், எங்கேயும் மார்க்ஸ் கட் பண்ணக்கூடாதுன்ற ஒரு ஐடியா வரணும்னா யூ ஹாவ் டு ட்ரா டயக்ராம்ஸ். ஸோ அந்த இன்ஸ்ட்ரக்ஷன்ஸ்லேயே கொடுத்துருப்பாங்க, ட்ரா டயக்ராம்ஸ் வேரெவர் நெசசரி. ஓகே, தட்ஸ் வெரி 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 இம்பார்ட்டன்ட். ஸோ முடிச்சிட்டோம், 2 டயக்ராம் முடிச்சிட்டோம். இப்போ நெக்ஸ்ட் டயக்ராம் போலாம், ஓவியூல். So ovule என்றது என்ன பசங்களா? Can anyone tell me where is ovule present? It's very simple. It's present inside my ovary. கரெக்டா? என்னோட ovule குள்ள தான் என்னோட female gametophyte இருக்கு. And that female gametophyte practicalsக்கு tips கொடுங்க. Sure. I will tell you. Okay. So என்ன experimentக்கு tips வேணும்னு சொல்லுங்க. கண்டிப்பா அதுக்கு ஏத்த மாதிரி நான் சொல்றேன். So you have your embryo sac, which is your female gametophyte. என்னோட female என்ன சொல்றது, கேமெட் வந்து எம்பிரியோ சாக், ஓவியூல் கிடையாது. ஓவரிக்குள்ள என்ன இருக்கு? ஓவியூல் இருக்கு. ஓகே, ஓவியூல்குள்ள என்ன இருக்கு? எம்பிரியோ சாக் இருக்கு. And my embryo sac is the most important structure. We call it as a female gametophyte. ஓகே, புரிஞ்சதா பசங்களா? என்னென்ன protective layers இருக்கு? embryo sac அடுத்தது nucellus ன்னு ஒரு structure இருக்கு. It's not nucleus, it is nucellus. Okay, then outer to the nucellus you have integuments, outer integuments, inner integuments. Egg cell is the, yes, egg cell is the female gametophyte. Egg cell இல்ல, embryo sac. Embryo sac. அந்த embryo sac குள்ள female gamete இருக்கு. Yes, that's correct. Yeah. Isolation of DNA, slide prep of pollen grain, and how to draw TS of ovary. We will discuss. Okay, கொஞ்சம் அந்த molecular basis of inheritance வரும்போது உங்களோட doubts நான் clear பண்றேன். Okay, integuments இருக்கு, outer இருக்கு, inner இருக்கு, and it has two ends, a micropylar end and a chelazal end. இந்த micropylar endல ஒரு சின்ன ஒரு germ pore மாதிரி இருக்கும், ஒரு pore மாதிரி இருக்கும். ஏன்? Because that is going to help in the introduction of my pollen grain, pollen tube development க்கு அது யூஸ் ஆகும். Okay, and I have my hilum and funicle with which they will interact with my ovary. So this is my entire ovule. Okay, very very important பசங்களா. 5 மார்க்கு கேட்கலாம், 3 மார்க்கு கேட்கலாம், but it's a very important diagram. Okay, next, the most important diagram, that is development of mature embryo sac. So in the mature embryo sac எப்படி develop ஆகுதுன்னா, you have something called as megaspore mother cell. Okay, என்னோட இந்த இதுல chelazal endல I will basically have a megaspore mother cell. Okay, so I will just write and show you that this one megaspore mother cell undergoes meiosis. Okay, both meiosis 1 and meiosis 2 அது undergo பண்ணும். Okay, அது undergo பண்ணோடனே it is going to produce 4 cells. Okay, இந்த 4 cells நம்ம என்னன்னு சொல்றோம்னா megaspore tetrad ன்னு சொல்றோம். Okay, இந்த 4 megaspores-ல 3 megaspores தான் they become degenerate, and only one becomes functional. So இந்த 3மே degenerate ஆயிரும், they will become degenerated. Okay, and only this becomes functional. Okay, in the functional megaspore will now undergo division. Okay, you can see here the functional megaspore is what is going to undergo the division. எப்படிப்பட்ட division na ஒரு it will undergo 2 cell stage, okay, 4 cell stage and 8 cell stage. 2 4 8 இது வரைக்கும் it will undergo only nuclear division. There is no cytoplasmic division until your 8 cell stage. Okay, 8 cell stage ஆனோடனே they will undergo a cell wall formation. Okay, அதான அப்புறம் என்ன நடக்கும்? Cell wall formation நடக்கும். Okay, so இந்த cell wall form ஆயிதான் 8 cells. 8 cells ஆ 7 cells ஆ? It is actually, let's count. Okay, உங்க chelazal end, உங்க micropylar end. Okay, in the chelazal end you have 3 antipodals. Okay, நடுவுல ஒரு central cell இருக்கு. How many meiosis and mitotic division, ma'am? See, meiosis is 2. Okay, 2 meiotic division. Mitosis is 3. Okay, 2 cell, 4 cell, 8 cell. Clear? Clear, lucid? Can we continue? Okay, so micropylar endla 2 synergids. Okay, one egg cell, and I also have 1 central cell. So totally it is 7 cell pasangla. Okay, ஆனா நியூக்ளியஸ் வந்து 8 நியூக்ளியஸ். எத்தனை நியூக்ளியஸ்னு கவுண்ட் பண்ணுவோமா? 1 2 3 4 5 6 7 8. So my mature embryo sac is 7 cell 8 nucleate stage. I want everyone watching the live to put it in the chat box. Embryo sac எப்படி இருக்கு பசங்களா? 7 செல் இருக்கு, 8 nucleus இருக்கு. So எந்த செல்ல வந்து 2 nucleus இருக்கு? Can anyone tell me in which cell do I have 2 nucleus? It is in my central cell. Okay, out of all the cells, my central cell is diploid in nature. Okay, இது மட்டும் தான் diploid. மிச்ச எல்லா செல்லுமே haploid in nature. Very good, Sam. Yes. Okay, so மொத்தமா நம்ம embryo sac எப்படிப்பட்ட ஒரு embryo sac? It is 7 cell. இது ஒரு MCQ type question வரலாம். 8 nucleate stage. Okay, so the entire development can come for 5 marks. Okay. Ma'am, முன்னாடி பார்த்த ஸ்லைட்ல chelazal end was downside, but here it is upside. Yes, அது வந்து டயக்ராமுக்காக இப்படி கொடுத்திருக்காங்க, அவ்வளவுதான். Okay, yes, try to remember it this way. இந்த பக்கம் பார்த்தீங்கன்னா இங்க பாருங்க, ஏன் மைக்ரோபைலார் எண்ட் இங்க இருக்குன்னு சொல்றேன். Because my pollen tube will come through my micropylar end and my pollen tube will actually enter through my synergids. That is why micropylar end is downwards here. Okay, புரிஞ்சதா? இப்படி ஞாபகம் வச்சுக்கோங்க. இங்க வந்து pollen tube development ஆகும், அதனால micropylar end is towards the other end. Okay, yes, very clear. இன்னொன்னு, இந்த micropylar endல filiform apparatus ன்னு ஒன்னு இருக்கு. யாருக்காவது filiform apparatus ஓட function தெரியுமா? Does anyone know what is the function of filiform apparatus? At least one of you in the class. Yes, it is going to guide my pollen tube. என்னோட pollen tube is going to be guided into my embryo sac via my filiform apparatus. Very good, very good, excellent. It guides the entry of pollen tube. Superb. So யாரெல்லாம் live பார்த்துட்டு இருக்கீங்களோ, please do like the live. Okay, அதுக்கு அடுத்தது, yes, paves the way for gamete. Superb, super. Signal to pollen tube. Very good, very good. எல்லாரும் சூப்பரா ஆன்சர் பண்ணிட்டு இருக்கீங்க. It guides the pollen tube. Very good, very good. Next, I have monocot and dicot embryo. இதுல ரொம்ப முக்கியம் என்னன்னா, it is your monocot. So இது வந்து உங்களோட monocot embryo. What is the difference between monocot and dicot embryo? Can anyone tell me? யாராவது சொல்றீங்களா? monocotனா என்ன, dicotனா என்ன? See, dicot means they have 2 cotyledons, correct? So here you can see one cotyledon and other cotyledon. Okay, and you have an embryonal axis. இந்த லைன் தெரியுதா? இந்த லைன் பேர் என்ன சொல்றோம் நம்ம? Embryonal axis ன்னு சொல்றோம். Okay, so this embryonal axis divides the cotyledon into 2. One is your one is ரெண்டு cotyledons இருக்கும், and above the embryonal axis, the region above the embryonal axis, இந்த region வந்து நம்ம epicotyle ன்னு சொல்றோம். And the region below the embryonal axis, very good, and the region below the embryonal axis is called hypocotyle. Okay, இவ்வளோ எழுதி நீங்க just label பண்ணீங்கன்னா போதும், you will get, it's usually diagram based questions will be for 3 mark or 5 mark. Okay, so this is your dicot embryo. ஆனா monocot embryo தான் ரொம்ப ரொம்ப முக்கியம். Okay, they have asked it in the PYQs also. So இந்த scutellum ன்னு ஒன்னு இருக்கு. மேம், இங்க cotyledon label பண்ணலையே, ஏன் மேம்? Because the scutellum is called as the cotyledon. The cotyledon of your monocot embryo is your scutellum. Okay, புரிஞ்சதா பசங்களா? மேம், cotyledonனா என்ன மேம்? கொஞ்சம் ஷார்ட்டா. Okay, fine. Cotyledon is a reserve food material. Okay, உங்க endosperm இருக்குல்ல, அந்த endosperm வந்து they basically are persistent in certain seeds. That is what we call it as cotyledon. உங்களோட ground nut எடுத்துக்கோங்க. அந்த ground nut ரெண்டா உடைச்சிங்க, ரெண்டா உடைக்க முடியுது, கரெக்டா? It is made up of 2 cotyledons. இந்த பக்கம் ஒரு cotyledon, இந்த பக்கம் ஒரு cotyledon. So that's what we call it as dicots. Yes, clear? Hi Tarun, hi. புரிஞ்சதா? ஹரி தமன். Yes, yeah. நான் சொல்லிட்டு இருந்தது, the cotyledon of your monocot embryo is called as scutellum. Okay, here you have coleoptile and coleorhiza. The region above the embryonal axis is called coleoptile, and the region below the coleoptile, okay, or the embryonal axis is called coleorhiza. ரொம்ப ரொம்ப முக்கியமான விஷயம். And can anyone tell me what does plumule and radicle develop into? Plumule develops into one and radical develops into one more, right? So plumule develops into shoot and radical develops into root. Simple. Okay, இதுதான் அதோட இது. அதே மாதிரி இங்கேயும் இருக்கு பாருங்க. Shoot apex and I have my radicle. Shoot apex நம்ம என்னன்னு சொல்றோம்? Plumule ன்னு சொல்றோம். Okay, so monocot embryo is very, very important pasangla. And this root cap is covered by coleorhiza. Okay, similarly, your shoot apex is covered by coleoptile. Yes, shoot and root. Exactly. You can go GK version. You can go check the live section. Okay, sir would have updated his classes there. Okay. This is the diagrams கேட்க மாட்டாங்க. Okay, I'm very sure they will not ask you to draw this diagram, but it is very important for diagram based questions. Diagram based questions க்கு இது ரொம்ப important. அதனால தான் நான் include பண்ணேன். Okay, so here you can see it's a dicotyledon. Okay, here you can see a monocotyledon, and this is your maize. எல்லாருமே corn பார்த்திருக்கீங்களா? Sweet corn. Sweet cornல இன்னும் clear-ஆ தெரியும் அந்த region. Okay, if this is your sweet corn பசங்களா, you can see a white region here, correct? And the white region will be covered by a yellow region. Have you guys observed it? So in the white region is your embryo, okay, and the yellow region is your endosperm. Okay, புரிஞ்சதா? இந்த embryoக்குள்ள தான் உங்க coleoptile, plumule, radicle, coleorhiza அதெல்லாம் இருக்கு. So this is the structure of your maize. Okay, then I have my false fruit. Can anyone tell me what is false fruit? True fruits, false fruit in irukku. True fruit na enna, false fruit na enna? Quickly let me see. How many of you will tell me? True fruits and false fruit. The one which is given here is a false fruit. Okay, solunga. True fruit na, they develop from ovary. True fruit enga irundha develop? Yes, fruits that develop from thalamus is called false fruit. Okay, idu vandu they develop from thalamus. Okay, example enna? Apple, strawberry, cashew nut. All these three are very good examples for your false fruit. Okay, neenga apple la saapringala, it is nothing but your thalamus. Okay, yes, it is called false fruit. And in the mari questions idhula irundhu kekkalam. In the diagram kodutittu they can name it as x and y. They can ask you to identify what is this x and y. That kind of questions can be asked. Yes, apple and strawberry. Banana false, it's true fruit. Banana is a true fruit. Okay, இப்போ male reproductive system, we have moved on to reproductive system. So male reproductive system oda parts romba mukkiyam. Okay, and the questions can be asked on the testis. Okay, and what is the pathway through which the sperms travel, that can also be asked here. Okay, yes. Then female reproductive system, again a very, very important question. Okay, so what you need to understand here is, you need to understand there are a pair of ovaries, there is uterus, and the uterus has 3 layers. Okay, that is your perimetrium, outer perimetrium, parthenocarpy and okay, very interesting question. சொல்லுங்க பசங்களா, parthenocarpy na enna, parthenogenesis na enna? See, parthenocarpy is the development of fruits without fertilization. Okay, like you guys said, banana la or parthenocarpic fruit irukku. Parthenogenesis is the development of an organism without fertilization, like your honeybees. Okay, yes or no? Yes or no? All of you understood parthenocarpy and parthenogenesis? Parthenocarpy is in plants, parthenogenesis is in animals. Okay, so honeybee la enna avo, you have an haploid gamete which will develop into male, and this is called as parthenogenesis. Okay, purunjada? Everyone understood the difference between parthenocarpy and parthenogenesis? Someone asked, I think it was lucid. Yeah, is it clear? Okay, இப்போ நம்ம female reproductive system குள்ள போகலாம். Okay, it has a cervix, cervical canal and vagina, and this you have your ovary which is held in position by your fimbriae. Okay, adukapro you have 3 important regions of your fallopian tube. Fallopian tube நம்ம oviduct ன்னு கூட சொல்றோம். Okay, and fertilization happens in the ampulla. எங்க நடக்குது பசங்களா? Ampulla of your fallopian tube or oviduct. So the sperm has to travel till the ampulla. Okay, so diagram பாத்தீங்கன்னா இந்த எல்லா விஷயமும் நீங்க புரிஞ்சுக்கலாம். And where does the embryo go and implant? Does anyone know where does the embryo go and implant? The embryo goes and implants in the uterine wall. Okay, and the shedding of endometrium is called menstrual cycle. Okay, yes, very good. Just by seeing one picture ellame ungalku nabagam vandirum. Okay, so this is just not a diagram, important diagrams matrum illa, important question session kuda vachikalam. Next we have the sectional view of seminiferous tubule. So where is this seminiferous tubule? Yes, very good Lucid, very good Sam Tamilan. Seminiferous tubules enga irukku pasangala? It is present in my testis. Okay, and here in the seminiferous tubule avlo mukkiyona, it is the one which is going to be involved in spermatogenesis. Okay, the part of your testis which is involved in your என்ன சொல்றது, the spermatogenesis is your seminiferous tubules. Okay, உங்க seminiferous tubule oda section is very very important. இது ஏன் நான் திருப்பி important ன்னு சொல்றேன்னா, they are going to be asking you certain questions. They can label it and they can ask you what is their function. They can label it and they can ask you to identify it. Okay, அந்த எல்லா விஷயமும் இங்க நடக்கலாம். Okay, so you have your spermatogonia. These spermatogonia are called the male germ cells. Okay, and these male germ cells will undergo mitotic division to form primary spermatocyte. Then primary spermatocyte will undergo meiotic division to form secondary spermatocyte. Now the secondary spermatocyte will undergo another meiotic division to form spermatid. Now the spermatid will develop into sperms. Okay, that we will see as a separate picture in spermatogenesis. Okay, it is the sectional view of seminiferous tubule. I have a question. Can anyone tell me what is the function of Sertoli cell? Sertoli cells oda function yarad solla mudiyuma? Ma'am, idiliye we have in practice to Viva la what questions they can ask. They can ask. That's what I'm asking you. Function of Sertoli cell kepanga. எத்தனை meiosis நடக்குதுன்னு கேட்பாங்க. And the mari they can ask. Okay, very good, very good. They provide nourishment to what? They provide nourishment to the developing sperms. Okay, yes, very good, excellent, excellent. Ipo structure of sperm, again an important 5-mark question. Okay, sperm oda diagram eludi adoda parts explain பண்ணுங்க. What are the 3 parts of the sperm? It has a head, it has a 4 part இருக்கு. Okay, ஒன்னு வந்து head, neck, okay, middle piece and tail. Okay, modalla what is there in? Yes, very good, Shubhlakshmi. Yes, என்னோட ஹெட்ல என்ன இருக்கு பசங்களா? My head has plasma membrane, it has nucleus and acrosome. இந்த acrosome base பண்ணி question கண்டிப்பா கேட்கலாம். Does anyone know what is acrosome? Acrosome na என்ன? Anyone? It is actually பசங்களா a modified Golgi apparatus. என்னது? Modified Golgi apparatus. ரொம்ப முக்கியமான விஷயம். So what is this? Yes, what is it actually going to contain? Golgi apparatus ன்னு சொல்றீங்க மேம், அதுல என்ன இருக்கும் மேம்? It contains various lytic enzymes. Okay, and these lytic enzymes are going to help in breaking the egg membrane and penetrate into the egg. Yes, it helps to penetrate into zona pellucida. Very good. It secretes enzyme. Very good, very good. அவ்வளவுதான். Okay, இதுல இருந்து ஒரு புது விஷயம் கத்துக்கிறீங்க, கரெக்ட்டா? Then neck is very short. Then I have my middle piece. The middle piece has mitochondria. Yeah, mitochondria. Why is mitochondria present? Because it is going to provide the energy for the motility of the sperm. Okay, because the sperm has to swim inside the female reproductive system. அதுக்கான energy தேவை. That is provided by mitochondria. Okay, and what is tail? This tail is actually going to help in movement. It is an undulating movement. The kind of movement in sperm is undulating, whip-like movement in kuda solluvom. Okay, yes. Can we continue pasangla? Is structure of sperm clear? Can we continue? Can I get green hearts in the chat section so we can continue? Yes. Spermatogenesis and oogenesis. 100% sure short question pasangla. நீங்க இது வந்து டயகிராமா பெருசா ஒண்ணுமே கிடையாது. இதுல ரொம்ப நீட்டா வரையணும் அப்படியெல்லாம் ஒண்ணுமே கிடையாது. It has to be logically correct. Okay, so now quick spermatogenesis and oogenesis explain பண்ணிடுறேன். Okay, see, spermatogenesis starts only at puberty. Okay, but oogenesis starts when the female child is in the fetal stage itself. அதுதான் முதல் difference. Okay, so as we already discussed, the spermatogonia undergoes mitosis to become what pasangla? Primary spermatocyte. Now this primary spermatocyte undergoes 2 meiotic division. first meiotic division ஆனோனே secondary spermatocyte form ஆகும். Second meiotic division ஆனோனே spermatid form ஆகும். The spermatid now undergoes differentiation to produce spermatozoa. Spermatozoa is also called sperm. That's it. It's very easy. இங்கதான் இருக்கு confusion. நிறைய பேருக்கு oogenesis confusing இருக்கும். What are the things that you people get confused? முதல்ல oogenesis எப்போ ஸ்டார்ட் ஆகும்? It starts in the fetal life. Fetal stageலயே ஸ்டார்ட் ஆகும். ஒரு குழந்தை, ஒரு girl child வயித்துல இருக்கும் போதே oogenesis ஸ்டார்ட் ஆகும். Okay, but it is arrested in the first meiotic division முன்னாடி அது arrest பண்ணப்படுது. arrestனா என்ன? stop பண்ணப்படுது, inhibit பண்ணப்படுது. Okay, and when does it again start? It starts at puberty. Okay, it starts at menarche. எப்போ அந்த menstrual cycle start ஆகுதோ அப்பதான் அந்த meiotic division resume பண்ணப்படுது. எந்த meiotic division? First meiotic division. Okay, so you can see at the puberty you have a secondary oocyte that is produced and you have first polar body that is produced. இங்க வந்து ரொம்ப முக்கியமான விஷயம் பசங்களா, ஒரு இதுல ஊகோனியால இருந்து ஒரே ஒரு ஓவம் தான் ப்ரொடியூஸ் ஆகும். ஆனா இங்க அப்படி கிடையாது. ஒரு ஸ்பெர்மாட்டோகோனியால இருந்து 4 ஸ்பெர்ம் ப்ரொடியூஸ் ஆகும். Okay, you can make a table difference between spermatogenesis and oogenesis. Compare பண்ணி படிச்சீங்கன்னா ரொம்ப ஈஸி ஆயிடும் படிக்கிறது. Okay, and when does the secondary oocyte undergo second meiotic division? Can anyone watching the live tell me? The second meiotic division எப்போ வந்து complete ஆகும்? First meiotic division எப்போ complete ஆகுது? During puberty. அது என்ன மேம்? polar body. Very simple. இது ஒரு இதா actually it should produce 2 equal cells. கரெக்டா? ஆனா இங்க வந்து நடக்காது. இப்படி ஒரு பெரிய செல் ஃபார்ம் ஆகும், ஒரு குட்டியான செல் ஃபார்ம் ஆகும். இந்த போலார் பாடி டீஜெனரேட் ஆயிரும். இந்த போலார் பாடிக்கு எந்த ஃபங்க்ஷனுமே கிடையாது. Okay, that's how it works. Yeah, very good, very good. This second polar body and your ovum is produced only during fertilization. After fertilization கிடையாது. எப்போ வந்து உங்க sperm வந்து உங்க egg enter ஆகுதோ அப்பயே the second meiotic division gets activated in my egg. Okay, and it is going to get converted into ovum. Yes, very good, very good. So என்ன நடக்குது பசங்களா? ரொம்ப முக்கியமா புரிஞ்சுக்கோங்க. Yes, I will write it also if you guys want. The meiotic division begins in fetal stage. எப்போ begin ஆகுது? Fetal stage. Okay, to be precise, I will write first meiotic division. Okay, it is completed. எப்போ complete ஆகுது? Completed during puberty. And the second meiotic division okay, is completed only during fertilization. எப்போ fertilization நடக்குதோ அப்போ மட்டும்தான் இது complete ஆகுது. Okay, clear பசங்களா? Can we continue? Yes or no? Is oogenesis and spermatogenesis clear? Yeah. Now, what is Graafian follicle? I see someone writing Graafian follicle. Yes, Graafian follicle is a mature follicle. Okay. Yeah. இது புரிஞ்சிச்சுனா now we can go into molecular basis of inheritance. Okay, so மிச்ச சில diagrams இருக்கு உங்களோட இதுல implantation காமிக்கிற ஒரு diagram, அப்புறம் development of Graafian follicle காமிக்கிற ஒரு diagram இருக்கு. அது வந்து diagram-ஆ கேட்க மாட்டாங்க, but diagram based questions-க்கு கண்டிப்பா கேட்கலாம் அந்த questions. Okay, now let's move on to molecular basis of inheritance. So molecular basis of inheritance la romba mukkiyamaana vishayamna double helix model of DNA. Here you need to mention certain things, okay, like your diameter. So what is the diameter of a double helix DNA pasangla? Can anyone tell me what is the diameter? It is 20 angstrom. Okay, and the number of bases per turn, it is 10 base pairs. Okay, then I have it is your Rise is 3.4 nanometer. Ma'am, rise na enna ma'am? ஒரு base pair க்கும் இன்னொரு base pair க்கும் இருக்க distance தான் நம்ம rise per base ன்னு சொல்றோம். Okay, these all things you need to write and you need to mention it. Yes, very good. It is 10 base pairs. Okay, so if you write this much, very rarely they ask double helix model of DNA or Watson-Crick model of DNA, but this is very, very important to understand for writing answers. Okay, இப்போ நம்ம further போலாம். Nucleosome. Can anyone tell me what is nucleosome? Very good, very good. What is nucleosome, pasangla? Can anyone tell me? What is this nucleosome composed of? Nucleosome is composed of DNA and a protein called histone. Okay. So, ஒரு செல்லிலும் pasangla, they are positively charged histone. Very good, very good. ஒரு செல்லிலும் there is this 2.2 meter long DNA. In the 2.2 meter long DNA is basically packed in a very minute space inside your cell, inside your nucleus. So, அந்த packaging வந்து ஒரு protein தேவைப்படுது, அந்த protein வந்து histone. Correct? DNA and protein octamer. Very good, very good. So, histone வந்து எப்படிப்பட்ட protein? Positive. DNA வந்து என்ன charge? Negative. Okay. So, the histone will form an octamer and you will have the DNA that is surrounding your octamer. Okay. So, இதை நம்ம என்னன்னு சொல்றோம்னா beads on a string ன்னு சொல்றோம். Beads on a string. Okay. Can anyone tell me how many base pair? Octamer na 8 protein இருக்கு. It has 8 proteins. Okay. What are the proteins? H2A, H2B, H3 and H4. Okay, இந்த ஒரு புரோட்டீனும் ரெண்டா இருக்கு. Okay, that will be octamer. Now clear? Harithanan. Yeah, Taman, I'm correct. Harithaman, is it clear? So that is what we call it as octamer of protein. Yeah, I had one question. ஒரு histoneல, okay, or one nucleosome consists of how many DNA base pairs? எத்தனை DNA base pair இருக்கு? யாருக்காவது தெரியுமா? Does anyone know? A nucleosome consists of 200 base pairs. இது உங்க NCERT bookலயே இருக்கு. If you want, you can open and check. Okay? Yes, 200 base pairs. Very good, very good. So, nucleosome could be a potential 5-mark question. Okay? Histone is positively charged and DNA is negatively charged. And can anyone tell me what is responsible for the positive charge of histone? There are two. Yeah, okay. I'll call you Hari. Yes, that's easier. Okay. Yeah. Can anyone tell me what are the 2 amino acids that's present in your histone? Hi. Vishnu Priya, hi. Anybody? Let me write. Okay. Lysine and arginine. Idella MCQ question, neat question. Okay, lysine and arginine are the 2 important amino acids that's present in your histone. Okay, yes. Hi Sindhuja, hi. So in the nucleosome patti information, if they ask you to write the nucleosome model, please do write this very easy diagram. Okay, can I continue? Is nucleosome clear? இப்போ நம்ம Hershey-Chase experiment பத்தி பார்க்கலாம். I think all of you know what Hershey-Chase do, right? Why did Hershey-Chase conduct all these experiments? ஏன் வந்து அவர் connect பண்ணாரு? Remember what? Remember this practice. Listen more, write more, practice more. Okay? நிறைய வாட்டி கேளுங்க, நிறைய வாட்டி எழுதுங்க, அதை பார்த்துட்டே இருங்க, ஈஸியா ரிமெம்பர் பண்ணிடலாம். ஹாய் சுமிதா, ஹாய். இப்போ ஒரு question கேட்டேன். Hershey-Chase எதுக்கு இந்த எக்ஸ்பெரிமெண்ட் பண்ணாங்க? It was used to prove DNA is the genetic material. Okay, இது ரொம்ப இம்பார்ட்டன்டான எக்ஸ்பெரிமெண்ட் and this diagram is also very important. Diagram based question க்கு கேட்கலாம், இல்ல அந்த எக்ஸ்பெரிமெண்ட் கேட்கும்போது obviously very good to prove that DNA is the genetic material. இவங்க தான் அந்த ஃபைனல் ப்ரூஃப் கொடுத்தாங்க. உங்க டெக்ஸ்ட் புக்ல 3 எக்ஸ்பெரிமெண்ட் இருக்கு. Griffith experiment, Oswald experiment and your Hershey-Chase experiment. இந்த Hershey-Chase experiment நம்ம blenders experiment னு சொல்றோம். So இதையும் மார்க் பண்ணிக்கோங்க. இது ரொம்ப முக்கியமான ஒரு விஷயம். Hi Sam, hi. Okay, can we continue pasangla? Then Coming to the most important thing, Michelson and Stall experiment. Okay, all these I have done full short videos. Okay, you can go and check it out in the video section. Molecular basis of inheritance. This ellame clear explain pannirken. Okay, so you have this. Why was this done? Can anyone tell me why was Michelson and Stall experiment done? What was the reason, or what did they prove? They proved that DNA replication is semi-conservative. So DNA replication semi-conservative ன்னு சொல்லி prove பண்ணதான் இவங்க வந்து இந்த experiment பண்ணாங்க. Yes? Yes, very good. இது ரொம்ப முக்கியமான question. Okay. And what did they use? They use N15, heavy nitrogen. Okay. இங்க இவங்க என்ன யூஸ் பண்ணாங்க? இது ஒரு ஷார்ட்டான MCQs. Here they used P32 and S35. P32 na enna? Phosphorus 32 and S35, sulfur 35. Yes, they used E. coli. Inga vandu E. coli da use pannanga. Okay, so these two experiments I have done in detail. You can go and check it out. Sulfur and phosphorus. Yes. Next important, replication. Right, replication. Oh, sorry. Okay, so replication pati what does this indicate? This indicates a replication fork. Okay, and when is this replication fork formed? When a DNA helicase, enna idu pasangala, DNA helicase goes and binds to the ORI. Okay, can anyone tell me what is the function of helicase? Helicase or a function enna pasangala? Yes. Anyone? Helicase sort of function, it is going to unwind my helix. Okay, when it unwinds my helix, there will be a fork-like structure that will be formed, and that fork-like structure is called as replication fork. The Y-shaped structure or the fork-like structure that is formed because of the action of helicase. So in the helicase, enna pannona, it's going to break the hydrogen bonds. Okay, and as a result, the two strands will separate. Next, you'll have the synthesis. One strand will be synthesized from 5' to 3'. Okay, that is called a continuous strand, and the other strand will be synthesized in the opposite direction. Adha vandu nama discontinuous strand nu solrom. Okay, so your DNA replication is semi-conservative. Okay, semi-conservative and semi-discontinuous. So when they ask you replication as a 5-mark question, you can use this diagram. Yeah, thank you so much. Yes, so semi-conservative and semi-discontinuous. Romba mukkiyam. So please, DNA ki nadula hydrogen bonds are there, not nitrogen base. Yeah, hydrogen bonds are there. Yes, the hydrogen, ipa parunga, A, T, G, C. Ipa eppadi irukka? Okay, in the helicase add pandroma. In the helicase enna pannom, it will break the hydrogen bond and it will separate the two strands. Now it is clear, Hari? Is it clear? And those two separated strands will form a Y-shaped structure. Yeah, so when they ask DNA replication, please do use this diagram. And you should also mention about SSB protein, single strand binding protein, which is going to prevent the rewinding of the helix. Okay, and which is the important enzyme that is involved in DNA replication? Can anyone tell me which is the most important enzyme that is involved in DNA replication? Or enzyme irukku, hero of DNA replication? Yes, what is the hero, or who is the hero of DNA replication? It is your DNA polymerase. Okay, nama almost full syllabus or what revise pannittu irukom. Okay, I thought of doing only diagrams, but it doesn't work that way. Yes, so your DNA polymerase, RNA polymerase and the transcription, DNA polymerase and the replication. Okay, can I continue? Now let's talk about transcription. So transcription in prokaryotes da unga syllabus la irukku. It is a very important 5-mark question. 3 steps irukku: initiation, elongation and termination. Okay, so what happens in initiation? Initiation la enna avuna, I have the hero of my transcription. Transcription oda hero yaru? RNA polymerase. So in the RNA polymerase enna pannona, it goes and binds to a specific site on my DNA, and this site is called promoter. என்னோட promoter க்கு போய் அது bind ஆகும். Once it goes and, yes, very good, very good. Once it goes and binds to that, you will have my initiation of my transcription. அது கூட ஒரு helper இருக்காங்க. அந்த helper பேரு sigma factor. Okay, hero, side hero, second hero இருப்பாங்கல்ல. மொத hero வந்து யாரு? RNA polymerase. Second hero யாரு? Sigma factor. இந்த sigma factor நம்ம என்னன்னு சொல்றோம்னா initiation factor ன்னு சொல்றோம். Okay, this sigma factor helps in recognition of promoter. என்னோட promoter recognize பண்ண இந்த sigma factor ரொம்ப ரொம்ப உதவும். Okay, can I continue பசங்களா? இப்போ என்ன நடக்குது elongationல இந்த RNA polymerase, it's one man army. It has helicase activity. It is going to unwind the helix. It is going to form a bubble like structure called transcription bubble, and you can see this yellow color structure, that is your RNA being synthesized. Okay, finally your, as in when your RNA polymerase reaches your termination site, இப்போ ஒரு இன்னொரு helper வராரு. அந்த helper பேர் என்ன? Rho factor. So your initiation factor is your sigma factor and your termination factor is your rho factor. Yes. Okazaki comes in replication, not here. Okay. Yeah, there is, I didn't get your question, Raghuram. Can you please repeat it? Okay. So initiation factor is sigma factor. Termination factor is rho factor. You will explain பண்ணி உங்க diagram neat எழுதி இதுக்கு ஒரு diagramக்கு ஒரு 3 point எழுதினீங்கன்னா போதும், you will be getting that full sure short 5 marks. Okay, next போலாம். This is your post transcriptional modification in eukaryotes. Very very important question. Okay, so என்ன நடக்கும்னா eukaryotes-ல quick-ஆ சொல்லிடுறேன். இது வந்து உங்க nucleus. Okay, உங்க nucleusல DNA will be converted into RNA, correct? And this RNA will be transported into your cytoplasm for translation. Okay, so since the RNA has to be carefully transported from the nucleus into the cytoplasm for translation, there will be certain post-transcriptional modification. There are 3 important post-transcriptional modification. Can anyone tell me? Yes, yes, yeah, correct. Yes, Hari, yes. You're just skimming through the topics. What you have is a very basic topic. So we can teach transcription for almost 3 hours. That is that huge. Transcription is that huge. And each and every topic is that huge. Okay, yes. So, 3 விதமான post-transcriptional modification இருக்கு. Capping, polyadenylation and splicing. Okay, so முதல்ல capping பத்தி பார்க்கலாம். To the 5' end. Capping எப்போ நடக்கும் பசங்களா? Only to the 5' end of my RNA. So, இதுதான் 5' endனா அங்க ஒரு ஸ்பெஷலான பேஸ், that special base is called as 7-methyl. Hi Deepak, hi. Yes, splicing, capping and tailing. Very easy. இது 3 மட்டும் ஞாபகம் வச்சுக்கிட்டீங்கன்னா போதும், ஈஸியா ஆன்சர் எழுதிடலாம். ஓகே, இந்த 7-methyl guanosine வந்து என்னோட 5' endக்கு ஆட் பண்ணப்படுது. அதை தான் நான் வந்து cappingன்னு சொல்றேன். ஓகே, tailingனா என்ன? To my 3' end. To my 3' end, I have poly A residues that is being added. Okay, and I have something called as splicing. So what is splicing? Anybody watching the live, can you tell me what is splicing? It is the removal of non-coding introns. Okay, so introns, exons in rendu irukku. So introns are non-coding, exons are coding region. Okay, the process of 200 to 300 adenylate residues. Yes, correct. So நான் இப்போ வெறும் என்ன சொல்றேன்னா, I'm teaching you only diagrams. So I'm just taking you through the topic. Yes, removing of introns. Absolutely correct. Okay. So இது 3 தான் ரொம்ப முக்கியம். அதுக்கப்புறம் translation. You should write this machinery. Okay. இன்னும் பெட்டரான picture வேணும்னா you can go and refer my translation video. Okay, I have dealt almost 45 minutes video பண்ணிருக்கேன் translation மேல. You can go and check it out. Okay. So what is translation பசங்களா? It is protein synthesis. It is a large part of your central dogma. Okay, protein synthesis நடக்குது. You have your ribosomes. என்னென்ன involved ஆயிருக்கு? Ribosomes involved ஆயிருக்கு, tRNA involved ஆயிருக்கு, mRNA involved ஆயிருக்கு. All the type of RNAs are involved in the synthesis of a protein. Okay, yes. Lacoperon, how can one forget lacoperon? Correct? So who discovered lacoperon? Anyone who discovered lacoperon? It was Jacob and Monod. இதெல்லாமே important questions. Okay, so ரெண்டு விதமான things இருக்கு. The operon is on. எப்போ வந்து operon on இருக்கும்? In presence of inducer. The operon is off when there is no inducer. இங்க inducer na யாரு? Who is the inducer? The inducer is lactose. Okay, so மூணு முக்கியமான region இருக்கு. ஒன்னு வந்து உங்க regulator or repressor. Okay, I have my promoter, operator and my structural gene. In the ZYA James monad. Okay, nice. Jacob and ஞாபகம் வச்சுக்கோங்க. Jacob and monad. Yes. But ஏதோ Jane ஞாபகம் வருதா? So remember it as Jacob. Okay. Yeah. So you can see what happens in absence of lactose. This repressor goes and binds to the operator. இங்க இருக்க RNA polymerase cannot move any further. As a result, the operon is off. So இத நம்ம என்ன சொல்றோம்? Negative regulation ன்னு சொல்றோம். Okay. அதே என்னோட lactose இருக்கும்போது the lactose goes and binds to my repressor. It makes my repressor inactive. As a result, the repressor cannot go and bind to the operator and my RNA polymerase which is bound to the promoter is free to move across the structural gene and it is going to produce all my என்ன சொல்றது proteins that is needed for the metabolism of lactose. Enna protein? Beta galactosidase, permease, transacetylase. Idu enna pannom? It is going to convert my lactose into glucose and galactose. Adha nadakkum. Okay, romba mukkiyamaana vishayam. Can we continue? Yes. Can we continue pasanga? Important sure short question lac operon alla. Okay? Yes. Lactose with repressor is called allolactose. Yes, absolutely. Actually, அப்படி சொல்ல முடியாது. இந்த lactoseன்றது it gets converted into allolactose. And this allolactose goes and binds to the repressor. Okay? And that is going to inactivate my repressor. அதுதான் நடக்கும். Yes. If this is clear, we shall continue. This is இது வந்து டயகிராமா கண்டிப்பா கேட்க மாட்டாங்க. This is related to your DNA fingerprinting. Okay, they can just ask you as an application based question. So இதுல யார் actual who is the real culprit, who is the real parent? By observing the bands you just have to find out. Okay, so DNA fingerprinting. Then genetics la பசங்களா genetics la பெருசா டயகிராம் ஒண்ணும் இல்ல. Pedigree ரொம்ப முக்கியம், crosses ரொம்ப முக்கியம். Okay, you can remember the monohybrid practice பண்ணனும் அது இப்ப எல்லாமே தெரியற மாதிரி தான் இருக்கும் எக்ஸாம்ல போய் ஃபுல்லா blank ஆயிடும் I have seen so many students who do so well in class but examல அந்த dihybrid cross பண்ணும்போது ஏதோ ஒரு mistake பண்ணி ratios தப்பு பண்ணிட்டு வந்துருவாங்க yes or no so you have to practice a lot monohybrid cross okay dihybrid cross and test cross monohybrid dihybrid test cross இருக்குல அது எல்லாமே ஃபுல்லா கிளியரா பிராக்டீஸ் பண்ணோம் ரெண்டு பெடிகிரி கொடுத்திருக்காங்க okay one is the autosomal dominant and other one is the autosomal recessive okay so coming week I will take a live exclusively on pedigree and genetic disorders okay yes yeah இது ரொம்ப ரெண்டு முக்கியமான விஷயம் hi Jab hi hi everyone hi we'll continue okay sickle cell anemia what is this sickle cell anemia what is sickle cell anemia can anyone hi Jab JSB okay hi Latika hi okay so sickle cell anemia ன்றது என்னன்னா can anyone tell me whether it's an autosomal recessive disease autosomal dominant disease blood becomes sickle shape okay very close to the answer அது blood RBC blood sickle shape மாறுமா RBC sickle shape மாறுமா it is basically an autosomal recessive condition okay the autosomal recessive condition la enna avuna it is your a fault in my hemoglobin gene என்னோட hemoglobin gene ல ஏதோ ஒரு mistake நடக்குது என்ன mistake நடக்குதுன்றது ரொம்ப முக்கியமான question இங்க பாருங்க இதுதான் என்னோட நார்மல் ஹீமோகுளோபின் ஜீன் என்னோட நார்மல் ஹீமோகுளோபின் ஜீன்ல ஆறாவது பொசிஷன்ல எத்தனாவது பொசிஷன்ல ஆறாவது பொசிஷன்ல குளூட்டமேட்னு சொல்லி ஒரு அமைனோ ஆசிட் இருக்கு ஓகே இதே என்னோட சிக்கல் செல் அனீமியால என்ன ஆகுதுன்னா இந்த A வந்து யூவா ரீப்ளேஸ் பண்ணப்படுது மியூட்டேஷன்னால okay and this GUG is now coding for another amino acid called valine okay simple GAG இருக்கு GAG என்ன கோட் பண்ணுது glutamate இந்த டயகிராமா கேட்க மாட்டாங்க but definitely you can have a question from this. Okay, but 𝐾𝐢𝐬 codes for valine. Okay, there is a change in one single base pair. Okay, so the biconcave RBC, RBC is now becoming sickle shaped. In the mari because of just change in one amino acid. See the significance of change in one amino acid. Okay, and this sickle shaped RBCs do not carry oxygen properly, and hence it is called sickle cell anemia. Okay, other thavara, this RBCs, if in the blood vessel in the mari RBC, it goes and forms clot. Correct? So it results in clotting, it results in anemia, it results in heart failure. Naraya vishயம் நடக்கும். MCQs la கேட்டாங்க in the sequence and order. Options பார்த்தா it was so tricky too. Just remember these two. இதை மட்டும் ஞாபகம் வச்சுக்கோங்க ஹரி. இதை ஞாபகம் வச்சுக்கிட்டீங்கன்னா போதும், ரொம்ப ஈஸி. ஸோ யாரெல்லாம் லைவ் பார்த்துட்டு இருக்கீங்களோ ப்ளீஸ் மேக் அ நோட் ஆஃப் இட். NCERT புக் பக்கத்தில் வச்சுக்கோங்க, இதை மட்டும் மார்க் பண்ணிக்கோங்க. இது கண்டிப்பாக கேட்கலாம். Either in your MCQ they can ask or either in your 2 mark or very short answers they can ask. Okay? Yes. Stanley Miller experiment. இந்த experiment இருக்கா? Do you guys have this experiment? I just wanted to confirm it once. Can anyone please confirm? இது எங்க வருதுன்னா evolution chapterல வருது. Is it there? Okay, this experiment was done to prove chemical evolution. Okay, எதை prove பண்ண யூஸ் பண்ணாங்க பசங்களா? chemical evolution prove பண்ண இந்த எக்ஸ்பெரிமெண்ட்டை யூஸ் பண்ணாங்க. ப்ளீஸ் கன்ஃபார்ம் பண்ணுங்க. இந்த ஸ்டான்லி மில்லர் எக்ஸ்பெரிமெண்ட் உங்க சிபிஎஸ்சில இருக்கா? டெக்ஸ்ட் புக்ல இருக்கு, பட் ஐ ஜஸ்ட் want to make sure that whether this is deleted or it's there. Okay. Yeah. So the chemical evolution na enna pasangla? It is nothing but before the life was formed. First it was all your biomolecules like amino acids, proteins. Irukunda nenikren. I think it is there. Okay. Proteins, amino acids, carbohydrates adellame produce ago. Adukapram you have your life that came into existence. So chemicals were first synthesized. Chemicals first, life later. Okay. Check பண்ணிட்டு சொல்லுங்க. என்ன நடக்குது பசங்களா? What did this Stanley Miller do? He basically created a pre-earth, primordial earth. பல வருஷங்களுக்கு முன்னாடி, பல millions of yearsக்கு முன்னாடி நம்ம earth எப்படி இருந்துச்சோ அப்படி ஒரு experimental setup வந்து அவர் create பண்ணாரு. Okay, rationalized bookல it's there. Thank you. Okay. Yeah. So இது ரொம்ப முக்கியமான எக்ஸ்பெரிமெண்ட். இது வந்து உங்க டெக்ஸ்ட் புக்ல இருந்துச்சுன்னா இந்த apparatus, Stanley Miller apparatus is very very important. He created a primordial environment. Why oxygen not used in the experiment? There is oxygen here, no? Waterல இருக்குல்ல oxygen. It is there, right? Yeah. So என்னென்ன பண்ணாங்கன்னா, first he used boiling water. Okay, and the boiling water the water will be converted into water vapor, and all this water vapor were collected in this huge spark discharge. Okay, inga vandu rendu electrode vachi electricity pass பண்ணாங்க. அப்ப என்ன ஆச்சுன்னா இந்த spark discharge ல 4 important ஆன elements இருந்துச்சு, இல்ல molecules இருந்துச்சு. Methane, ammonia, water and hydrogen. Okay, so when they pass this electric current, okay, at a very high pressure, what will happen pasangla? There will be formation of molecules, right? So all these molecules were condensed here. Condensing na enna? It is a conversion of gases into liquid and they were trapped here. Okay, in the Stanley Miller in the trapped and liquid paakumbodhu he could see many small biomolecules like amino acids, okay, small sugars, adella avarala paaka mudinjadu. So he proved chemical evolution. Okay, புரிஞ்சதா பசங்களா? So Stanley Miller experiment எதுக்கு பண்றாங்க? To prove chemical evolution. Yes, primordial earthல O2 gas இல்ல. O2 gas அங்க இருந்தில்ல. Even I will tell, O2 gas is not there. But O2 was in the form of water. Water vapor இருந்துச்சு. Water vapor இருந்துச்சு. And that's why I'm boiling this water to give water vapor. Right? Ma'am, why oxygen is not used in this experiment? Yeah, that's the reason. Okay? Homologous, analogous organ, sorry, analogous organ mention பண்ண மறந்துட்டேன் picture. இதுவும் diagram கேட்க மாட்டாங்க. Okay, this is again from your evolution. Can anyone tell me what is homologous organ and what is analogous organ? Homologous organ, they have the same origin. Listen to me very, very carefully. They have the same origin, they have the same structure, but different function. Okay, example, 4 limbs of mammals. See, we man also have 4 limbs, our hands. Cheetah also has 4 limbs, whale also has 4 limbs, bat also has 4 limbs, but each of us use it for different function. We use it for a different function. Cheetah uses it for running, whale uses it for swimming, okay, bat uses it for flying, right? Structurally same, functionally different. Romba simple ah solli tharana. Okay, homologous na enna? Structurally same, okay, functionally different, okay. Adhe analogous organ na enna theriyuma pasangala? Can anyone tell me what is analogous organ? Yes, structurally different, same function. Very good. எல்லாரும் ரொம்ப சீரியஸா படிச்சிட்டு இருக்கீங்கன்னு நினைக்கிறேன். Structurally, okay, different. Example, functionally same. Can anyone tell me the function example for analogous organ? Any example for analogous? Homologous organ leads to divergent evolution. Enna mari evolution pasangala? Divergent evolution. Whereas my analogous organs, Darwin's finches, very good, very good. Then thorn and tendrils, very good. Then wings of butterfly and wings of birds, correct? Very good. So this leads to convergent evolution. Okay, important difference between homologous and analogous. 3 marks, 2 marks. Assalta mudichirala. Yes, Darwin's finches. Okay, clear pasanga? Idu வந்து picture கண்டிப்பா கேட்க மாட்டாங்க, but application based கண்டிப்பா கேட்கலாம். Selection. Okay, ரொம்ப ரொம்ப முக்கியமான விஷயம். This also comes under evolution. Okay, 3 விதமான selection இருக்கு. Can anyone tell me what are the 3 types of selection? Anybody? Okay, directional selection. Then you have stabilizing. Then you have disruptive. The 3 da selection in the graphs romba mukkiyam. Okay, what is selection? Explain their types. Okay, selection na enna pasangala? The nature selects organism with certain phenotypes. Okay, so ipo or example solren, or white mouse irukku, or gray mouse irukku, correct? So let's take that as the extreme phenotype. If both the extreme phenotypes are selected, then we call it as disruptive. Rendume extreme phenotypes like this. You can see only the extreme phenotypes are selected. Okay, inga pathina the medium phenotype is selected, or middle gray black illa white illa gray color is selected, then that is called your stabilizing selection. Directional na only one phenotype is selected. Okay, so 3 types: stabilizing, disruptive, directional. Graph is very, very, very important. Okay, yeah, we shall continue. Like now we are going on to human health and disease. Evolution varkiyum mudichachi. Human health and disease paathitu irukom. Here life cycle of Malarian parasite. Hi Antony, hi. Yes, life cycle of Malarian parasite. Important 5 mark question. Model question, what causes malaria? Papa ethana different answers varudhu paakalam. What is the causative agent of malaria? Yes, anybody? Malaria is caused by Plasmodium vivax. Idu oru species. Malariae. Okay, and falciparum. The most severe type of malaria is caused by your Plasmodium falciparum. Okay? Yes, see. I expect panna answers vandittu irukku. They are spread by female Anopheles mosquito, but it is caused by your Plasmodium vivax. Okay? Padi book mela panniyachi within 1 hour. Yes, yes. Now, I'm not actually doing everything in detail. Important, romba romba important. This short varondradhu matram dhaan pannittu iruken. Yes. Yeah. இப்போ இந்த மலேரியல் பாராசைட்டுக்கு வந்து 2 ஹோஸ்ட் தேவை. ஒன்னு வந்து நான் human being, இன்னொன்று வந்து the mosquito. Okay, so it has two hosts, human host and mosquito host. Adoda good evening, good evening. So adoda paadi life cycle, yes, adoda paadi life cycle on the human beings nadakudhu, micham paadi is happening in your mosquito. You guys have to write this entire diagram and explain. Okay, avlo perfect ah irukanala thevilla, or alavukku teacher ku puriyra mari irundha podum. See, we as examiners, we don't expect very beautiful diagrams. Even teachers, we know what can a student draw. We don't expect very beautiful, romba alaga paakave alaga irukanondra avashyame kedaiyadu. I have already told you, just have to write logically correct labeled diagrams. Okay, no, you are not too late. I'm still half the way through the session. Okay, yes, this is malarial parasite. Structure of antibody romba romba mukkiyam. What is antibody? What is other name for antibody pasangla? Immunoglobulin. Okay, who is producing antibody? Who is producing antibodies? Yes, who is producing antibodies? Vel Murugan, நீங்க பார்க்கலாம், உங்களுக்கு யூஸ் ஆகலாம். Okay, you can just see, I'm revising the whole topic here. Yes, T lymphocyte, it is your B cells or B lymphocyte. Okay, so whenever your B cell encounters an antigen, your B cell gets differentiated into a plasma cell. That plasma cell will produce antibodies. So in the antibody la 4 protein chain irukku. Okay, it has 2 heavy chain and 2 light chains. Okay, and these chains are connected by indha mari theriyudilla, what are they? They are called disulfide bridge. You need to இப்போ ஒரு 3 marks கேட்டாங்கன்னா கண்டிப்பா நீங்க லேபிள் பண்ணணும். ஓகே, குட் ஈவினிங், குட் ஈவினிங், டைசல்ஃபைட் பாண்ட்ஸ். ஓகே, ரொம்ப ரொம்ப முக்கியமான விஷயம். And you can see this is the place where antigen binds. Antigen na enna? It's a foreign pathogen. Foreign pathogen binds to your antibody, and then the antibody is destroyed using your antigen is destroyed using your antibodies. So நிறைய விதமான antibodies இருக்கு. You have IgG, okay, IgA, IgM, and IgD, and IgE. Okay, all these are the antibodies which are present based on the heavy chain. In the heavy chain, vera maarumbodhu the type of antibodies keeps changing. That's it. Okay, romba mukkiyamaana diagram. Innoru mukkiyamaana vishayam, the life cycle of HIV virus. Okay, so what is HIV virus? Human immunovirus. Okay, what does this HIV virus affect? Okay, human being. Okay, so what cell does this HIV virus? கண்டிப்பா பார்க்கலாம் உங்களுக்கு ஒரு quick revision நடக்கும். Yes, yeah, what does this HIV virus affect in your body? It affects your T lymphocytes. In T lymphocytes, especially your TH cells. Okay, in the HIV virus என்றது ஒரு retrovirus. Yes, correct Sam. Immune cell. HIV virus என்றது ஒரு retrovirus. It has RNA as its genetic material. So இந்த RNA என்ன ஆகும்னா DNA வா convert பண்ணப்படும். Okay, எப்ப convert பண்ணப்படும்னா only when it enters your host. இங்க பாருங்க, the RNA is entering. Okay, there is a process called reverse transcription. இது வந்து என்னோட viral DNA, viral RNA, right? Viral RNA, and this viral RNA is getting converted into DNA. Okay, and this DNA is called cDNA. C here stands for complementary DNA. Okay, and the conversion of RNA to DNA enna process pasangla? Reverse transcription. And the enzyme used is what pasangla? Reverse transcriptase. Thank you Rahul AR. Thank you. It's all because of your support we have reached so far. Okay, yeah, so reverse transcriptase என்ற ஒரு enzyme இருக்கு, that is going to help in the conversion of RNA to DNA. Okay, அது நடக்குதா? அது நடந்தது என்ன ஆகுது? It is going to go and basically integrate with my host cell DNA. It's going to start producing new virus particle, and those new virus particle will go and attack my TH cell. So my body cannot produce TH cells and my immunity is suppressed. அதனால தான் நான் அதை வந்து acquired immunodeficiency syndrome ன்னு சொல்றேன். Right? HIV virus causes what? AIDS. இது ரொம்ப முக்கியமான question பசங்களா. Either they can ask you HIV or malarial parasite life cycle. Okay, let's move on to the next one. இது சும்மா கொடுத்த like what I thought is இந்த structure கொடுத்துட்டு நீட்டுக்கும் கேட்கலாம் இது. இந்த structure கொடுத்துட்டு what is the structure ன்னு கேட்கலாம். Okay, you have this comes under your drugs. This is your cannabinoid and this is your morphine. Yes. Okay. Now we have moved on to microbes in human welfare. Biotechnology இன்னும் நம்ம போல we are ready to do it. In microbes in human welfare, you have biogas plant. So what is biogas? Ma'am, in biology for 2 mark differentiation question, how many points should we write? 4 points. 4 to 5 points. நீங்க கரெக்டா எழுதுனீங்கன்னா எல்லா பாயிண்ட்டும் ஷியரா இருந்துச்சுன்னா 4 பாயிண்ட் போதும். ஏதோ ஒரு பாயிண்ட்ல ஏதோ ஒரு தப்பு பண்ணியிருந்தீங்கன்னா write 5 points to be on a safer side. Yes. So biogas, biotechல bioreactors இருக்கு. Yes. So biogas la we basically have it's a gas mixture of methane. Okay, sometimes you have hydrogen and water vapor. Okay, and sometimes hydrogen sulfide is also present. Okay, so that is your biogas. Biogas is produced by bacteria called methanogens. Methanogens produce biogas. Okay, in the plant வந்து நீங்க கரெக்ட்டா எழுதணும். Okay, what is the difference between gobar? They are same. Gobar gas and biogas is the same. Okay, so biogas, the gobar gas in ஏன் சொல்றேன்னா gobar in Hindi means cow dung. Okay, so the gas which is produced using cow dung is called biogas. H2S இருக்கு correct, hydrogen sulfide. Okay, next age pyramid. I think I've skipped biotechnology chapter. I will tell you what is important. So இது வந்து age pyramids இருக்கு, expanding, stable and declining. Okay, so this comes under organism and population. 3 விதமான pyramids இருக்கு. Expanding population na the people in pre-reproductive age வந்து அதிகமா இருக்காங்க. Okay, reproductive is less, post-reproductive is less. வயசானவங்க ரொம்ப கம்மியா இருக்காங்க. Okay, stable na the pre-reproductive and reproductive people are almost the same. Okay, declining na remember the pre-reproductive population is very less. அப்பனா in the near future the population can decline. அததான் நான் வந்து declining population ன்னு சொல்றேன். இதெல்லாம் அசால்ட்டா ரொம்ப ஈஸியா மார்க் வாங்கக்கூடிய ஒரு டாபிக் பசங்களா. What are age pyramids? Draw them. இத கொடுத்துட்டு mitosis is equational division. It is a type of cell division. Okay, these are your age pyramids. Okay, இப்போ நம்ம types of growth curve பார்க்கலாம். இது ரொம்ப முக்கியம். Okay, ரெண்டு விதமான growth curve இருக்கு. Can anyone tell me what are the growth curves? You have your exponential growth curve and logistic growth curve. Okay, the exponential என்றது இந்த மாதிரி J shaped. Okay, logistic என்றது S shaped இந்த மாதிரி. Okay, S shaped and J shaped. Very good. It is very important. In the equation ஆகட்டும் எல்லாமே ரொம்ப முக்கியம். dN by dt is equal to Rn here. So dN by dt is equal to Rn. What is R? It is intrinsic rate of natural increase. R is given by B minus D. அதே இங்க வந்தீங்கன்னா dN by dt is equal to Rn K minus N by K. What is K? Anybody? What is K? Mitosis is a process of cell duplication. Yes, it's cell division, that's all. Yeah. Can anyone tell me what is K here? K is the carrying capacity. என்னோட ஒரு இடத்துல ஒரு என்கிட்ட 2 கிலோமீட்டர் இடம் இருக்கு. அந்த 2 கிலோமீட்டர் இடத்துல 2 ஸ்கொயர் கிலோமீட்டர் இடம் இருக்கு. 2 ஸ்கொயர் கிலோமீட்டர்ல மேக்ஸிமம் எவ்வளவு ஆர்கானிசம் வாழ முடியும்? That is what we call it as carrying capacity. K. Okay, remember these two. And someone told something. Yeah, in your exponential it is assumed that the resources are unlimited. என்ன சொல்லுது இது? இந்த growth இப்படி continue ஆயிட்டே இருக்கும்னு சொல்லுது. But is it possible practically? No, it is not possible practically. Okay, but when the resources are limited, எல்லாருக்குமே உங்களுக்கும் சரி எனக்கும் சரி பிளான்ட்ஸுக்கும் சரி அனிமல்ஸ்க்கும் சரி எல்லாருக்குமே we all have limited resources. Correct? So we have to use that limited resources and grow. The same thing is happening here. இது நம்ம S-shaped curve ன்னு சொல்றோம். This is more practically applicable. Which one? Logistic. ரொம்ப முக்கியமான விஷயம். Okay? Yes. Then coming to population interaction. இது வந்து ஒரு டயக்ராம்னு சொல்ல முடியாது, ஒரு டேபிள் பேஸ்ட் கொஸ்டின் சொல்லலாம். what are population interaction? It's the interaction between biotic and biotic. Okay, it could be a positive interaction like your mutualism, negative interaction. Positive na enna, both of them are benefited. Negative na enna, they are affected. அவங்களுக்கு ஏதோ ஒரு கெட்டது நடக்குது, that is called negative. Negative is competition. Positive minus is predation. Positive minus is parasitism. Positive 0 is commensalism. Minus 0 is amensalism. Okay, these are all different kinds of population interactions. Okay, so இதுக்கு வந்து நீங்க examples கத்துக்கணும், உங்க NEET examக்கு ரொம்ப உதவும். Okay, out this quality class for free. Okay, we guys need your support, that's all. Okay, we have this biodiversity. இது கூட மேம் இதையே இன்க்ளூட் பண்ணிருக்கீங்க, இதுலயும் questions கேட்கலாம். எப்படி மேம் இதுல questions கேட்கலாம்? They can ask you which is the maximum number of vertebrates which is present. Vertebrates எது வந்து அதிகமா இருக்கு? Fishes. Okay, actually இங்க plantல தெரியாம இங்க fungi include பண்ணிட்டு இருக்காங்க. Okay, but which is the maximum number of plant? Angiosperm. Angiosperm is the most abundant plant. Okay, உங்க invertebrates most abundant எது? Insects. Okay, இதுக்காக தான் நான் வந்து இந்த ஸ்லைட் இன்க்ளூட் பண்ணேன், because MCQ க்கு கண்டிப்பா கேட்கலாம் இது. Okay, yes, this is one more thing. This is the species area graph. Species area graph. Okay, this comes in biodiversity and conservation. Okay, so என்ன சொல்றாங்கன்னா ரொம்ப ஈஸி. when the area increases, clear ஆ யோசிங்க, இப்ப ஏரியா அதிகமாயிட்டே இருக்கு, அப்ப என்ன ஆகும்? the number of species also increases. and the species க்கு வளர ஒரு இடம் கிடைக்குது, they are getting a place to continue their life. Okay, as the area increases, the amount of species also increases. ரொம்ப simple. Okay, when you see this is given by S is equal to CA square. இதே நீங்க log square, இந்த பக்கமும் log அந்த பக்கமும் log எடுத்தீங்கன்னா log S is equal to log C plus Z log A. Okay, this graph is quite important பசங்களா. species area relationship graph ன்னு கேட்கலாம். Okay, can we continue all of you? Yes or no? Yes, அவ்வளவுதான். இல்ல நான் biotechnology மட்டும் மிஸ் பண்ணிட்டேன்னு நினைக்கிறேன், but I will tell you what is very important. I'll take a new screen and tell you. Okay, சொல்லுங்க, பயோடெக்ல எது ரொம்ப முக்கியம்? First, you need to understand the action of restriction enzyme. Okay, so you have this restriction enzyme called 𝐃𝐢𝐬𝐼. Okay, so this 𝐃𝐢𝐬𝐼 basically has a recognition site called GAA-TTC, correct? GAA-TTC. Okay, it is going to cleave gel electrophoresis, plasmid diagram. Yes, so it is going to cleave at this point. They are going to produce sticky ends. Okay, இது ஒரு important diagram. Okay, this is one important diagram ன்னு சொல்ல முடியாது, ஒரு என்ன சொல்றது, ஒரு note. Okay, which is where you have to draw something. Okay, next you have the most important thing, that is your plasmid. Okay, so plasmid. And what plasmid you have in your book பசங்களா? என்ன plasmid இருக்கு? PBr 322. Okay, you have PBr 322. So it has an ORI site. Okay, it has 2 MCS, 2 multiple cloning sites. Okay, one is your ampicillin resistance. Okay, other one is your tetracycline resistant gene. Inga restriction sites varum, 𝐾𝐢𝐬𝐼 and the restriction sites you can just draw. PBr 322 is very important. அதுக்கு அடுத்தது gel electrophoresis diagram. Yes, so what is this gel electrophoresis? So நான் NEET தமிழ் சேனல்ல gel electrophoresis பத்தி ஒரு ஷார்ட்டான வீடியோ போட்டிருக்கேன். I think it will be around 12 minutes. You guys can go and check it out. In detail I've explained about gel electrophoresis. DNA amplification, gel electrophoresis. Separation. Okay, gel electrophoresis is separation of DNA. Okay, separation method, very good, to separate DNA based on their size. Okay, இங்க என்ன இருக்கும்? இங்க வந்து well இருக்கும். Okay, so remember smaller fragments. எப்படி ரொம்ப thin இருக்கவங்க they can run fast, ரொம்ப fat இருக்கவங்க they cannot run fast, correct? Same thing here. Larger fragments will be close to the well. Okay. Smaller fragment will be away from the well. They can move very fast. Same thing here. Okay, இதெல்லாம் என்னது பசங்களா? Fragments. Okay, so this is how your DNA. Ma'am, sorry, நான் உங்க question கேட்கல. It's okay, no problem. So DNA electrophoresis ஓட use என்ன? Separation of DNA. Based on what பசங்களா? Based on size. Okay, இது ஒரு important ஆன diagram. Next, ரொம்ப ரொம்ப முக்கியமான விஷயம். PCR, polymerase chain reaction, right? Yes, they move to the anode. Yes, correct. DNA is negatively charged, so it moves towards the positively charged electrode, which is your anode. Okay. Next, PCR na enna? What is PCR? It is an amplification technique. Amplification na enna? They are going to help in the multiplication of my DNA. What are the 3 steps in PCR? Anybody? Denaturation, annealing and extension. இந்த மூணுமே ரொம்ப ரொம்ப முக்கியமான விஷயம். Yes, to multiply DNA, to amplify DNA. So at one temperature, DNA is denatured. Denatured na enna? The 2 strands are going to separate like this. Okay, annealing la enna nadakkum? The primers go and anneal. Okay, and in extension, I have the synthesis that is happening. Okay, so what is the enzyme that is used in your PCR? Enzyme. So ingella temperatures vera irukum. Okay, that's why we call PCR as a thermocycler, because the temperature keeps changing. Okay, yeah. Taq polymerase, excellent. Ammu, correct. ஏன் நம்ம வந்து இதுல Taq polymerase use பண்றோம்? Why are we using Taq polymerase? Anybody? Why don't we use the normal DNA polymerase? Because சொல்லுங்க, Taq polymerase எதுக்கு யூஸ் பண்றோம்? Taq polymerase is used because இங்க வந்து நான் temperature, high temperature, 90 degree, 72 degree அந்த மாதிரி temperature மாறிட்டே போகும். Okay, yes, Taq polymerase can withstand high temperature. It's a thermostable polymerase. Okay, so அதுக்கான ஒரு hint கொடுத்துட்டேன். இந்த questionம் கேட்கும். Why is Taq polymerase used? Yes, it is active in high temperature. Superb. ஒரே ஒரு part இருக்கு last, fermenters. Okay, the fermenters diagram மட்டும் கொஞ்சம் பார்த்து படிச்சிருங்க. I'll just tell you what are the 2 types of fermenters. What are the 2 types of fermenter which is given in your textbook? Can anyone tell me what are the 2 types of fermenters which is given in your textbook? Sparged type and stirred type. Okay, and the 2 bioreactor oda diagram padichukonga, stirred and sparged. Absolutely correct. Okay, so by 76 minutes, we have almost revised revised in that solva na solli kuduthunalla solla maten. I have just revised, I have just taken you through what is all important. Okay, I want you guys to go back and study this. Ivlo padicha poduma ma'am, kandipa podum. It's more than enough. Okay, so இப்போ நீங்க 5 marks prepare ஆயிட்டே இருக்கும்போது you also parallelly prepare for 2 marks, 1 mark and MCQs. Okay, so that is also happening there parallelly. Important diagrams and diagram based question ku idu podum. Yes, thank you so much for this session. This session was very effective. Thank you so much. This means a lot. Please do leave your comments. Ore or help, please do leave your comments in the comment section, so many people can watch this video. So comments paathu da nariya per video paapanga. So please do put your comments in the comment section, so it will be useful to many of them watching it. So if there is any feedback, please do let me know. I don't know how did I miss this biotechnology. Okay, applications la perusa onno diagrams kedaiyadu. You have that insulin diagram. Okay, and the A chain, B chain adu kuda pannirren. So see, this is your A insulin la, this is your C, this is your B. இத வந்து நம்ம pro insulin ன்னு சொல்றோம். என்னன்னு சொல்றோம் பசங்களா? Pro insulin ன்னு சொல்றோம். This pro Vanasila Ai from Kerala. Thank you. Yes. Ecological pyramids, yes, that I have to do, right? Pyramid of number, pyramid of age. I thought okay, I will include that when I do that. Nadula hydrogen and yes. Okay, so இப்போ என்ன நடக்கும்னா என்னோட இன்சுலின்ல என்னோட மெச்சூர் இன்சுலின்ல there will be disulfide bond. இந்த C peptide வெளிய வந்துரும். இந்த A and B peptide மட்டும் தான் இருக்கும். Okay, இது ஒரு important diagram. Okay, yes. So this session is very, very important for diagrams and diagram based question. I won't tell I have finished the entire session. I won't tell all that. Okay, ரொம்ப முக்கியமான question. நீங்க already ஒரு அளவுக்கு படிச்சிருந்தீங்கன்னா இந்த session ரொம்ப யூஸ்ஃபுல்லா இருக்கும். Okay, thank you guys. Wishing you all a very, very happy Pongal. Okay, so Pongal holidays கொஞ்சம் utilize பண்ணுங்க. At least ஒரு 1 or 2 days நல்லா படிங்க because the exams are coming very, very close. Okay, yeah. Thank you, Pasangla. Take care. Bye. Happy Pongal, all of you. Happy Pongal.